This is the second of two key concept videos on the nervous system. In particular, this focuses on the synapses. In the video, we will look at the structure of synapses, the transmission of nerve impulses across synapses, and the effect of neonicotinoids on the insect nervous system. In the first of these videos on the nervous system, we saw how an action potential is carried along a neuron, but how is it passed from one neuron to another? The junction between two neurons is known as a synapse. The structure of the synapse is shown in this diagram here. This is the first neuron along which the action potential will be transmitted. There are many mitochondria in the presynaptic knob, as ATP is required for nerve impulse conduction. There are calcium ion channels found in the membrane of the presynaptic knob. These are another type of voltage-gated channel, and so are opened and closed by changes in membrane potential. These are vesicles containing a signalling chemical called a neurotransmitter. An example of a neurotransmitter is acetylcholine, found in cholinergic synapses. Here is the synaptic gap, the gap between the presynaptic neuron and the postsynaptic neuron. Receptors are embedded in the postsynaptic membrane. These are complementary to the neurotransmitter. When an action potential arrives at the presynaptic knob, the change in membrane potential causes the voltage-gated calcium channels to open and the calcium ions diffuse into the presynaptic knob. The influx of calcium ions causes these vesicles containing neurotransmitter to move towards and fuse with the presynaptic membrane. When vesicles fuse with the membrane, neurotransmitters are released into the synaptic gap through the process of exocytosis. Because there is a high concentration of neurotransmitter on the presynaptic side of the synaptic gap, the neurotransmitter molecules diffuse across the synaptic gap down their concentration gradient. When the neurotransmitter reaches the postsynaptic membrane, it binds to the complementary receptors. These receptors are the receptors of ligand-gated sodium channels. So, as the neurotransmitter binds, it causes the opening of the sodium channel, allowing the influx of sodium ions, leading to the generation of an excitatory postsynaptic potential, which is a temporary depolarization of the postsynaptic membrane. If the threshold potential is reached, an action potential is triggered in the next neuron, and so the impulse continues its journey. To prevent continuous synaptic transmission, the neurotransmitter is rapidly broken down by specific enzymes. For example, the neurotransmitter acetylcholine is broken down by acetylcholinesterase into acetate and choline. The choline is then taken back up into the presynaptic knob by facilitated diffusion to be recycled and used to make more acetylcholine. The acetate is metabolized. The removal and breakdown of neurotransmitters play a crucial role in the regulation of synaptic transmission and preventing overstimulation of the postsynaptic neuron. Neonicotinoids are a class of insecticide that work by targeting the nervous system of insects. They are called neonicotinoids because their chemical structure is similar to nicotine, a natural compound found in tobacco plants. Neonicotinoids act as neurotoxins that specifically bind to acetylcholine receptors in the postsynaptic membranes of the insect's nervous system. Once bound to the receptors, the neonicotinoids act as agonists, which means they activate the receptors. Unlike the neurotransmitter acetylcholine, which binds briefly and is then broken down by acetylcholinesterase, neonicotinoids bind more persistently to the receptors and are not broken down by acetylcholinesterase. The continuous stimulation overexcites the insect's nervous system, which disrupts the normal transmission of nerve impulses. The insect is unable to control its muscles and cannot carry out essential functions. This often results in paralysis and death of the insect. As a result of concerns about the impact of neonicotinoids on pollinators, the use of this class of insecticides has been restricted in many countries, including the UK, the EU countries, Canada, the United States, Australia and India. So in this video, we have seen how the synapse is a junction between neurons, how an action potential is a brief rapid change in electrical potential of neurons involving depolarization and repolarization. Voltage-gated channels are channel proteins in plasma membranes that open and close in response to changes in membrane potential, controlling the passage of ions. The threshold potential is the minimum membrane potential that must be reached to initiate an action potential. A neurotransmitter is a signaling chemical released from the presynaptic neuron into the synaptic gap, enabling the transmission of a nerve impulse to the postsynaptic neuron. The excitatory postsynaptic potential, or EPSP, is a temporary depolarization of the postsynaptic membrane caused by the influx of sodium ions through ligand-gated ion channels in response to the binding of neurotransmitter. Ligand-gated channels are channels in plasma membranes that open or close in response to binding of specific signaling molecules, known as ligands. Acetylcholine is a type of neurotransmitter that transmits nerve impulses across cholinergic synapses. And neonicotinoids are insecticides that attach to acetylcholine receptors in postsynaptic membranes of synapses. Note the highlighted words are our buzzwords, the words that you should try to use in your extended answers in your exams.